Hello everybody, and this is the introductory video to an experiment that I've been talking about with my students, and now with the quarantine and the coronavirus, I'm at home, so we can't do the experiment in the lab, sadly, but we can do it here. So we've talked about concentrations. Okay, and in chemistry we talked about molar concentration. We've talked about in another video about parts per thousand or parts per million, things like that. We also talked a little bit about that in APES as well. So the idea behind this experiment, you see these little green leafy things here. They look like mini lily pads. They are called duckweed. You do not find them in California, you find them in Texas. I have been culturing them ever since I got a small batch from an APES workshop, an AP Environmental Science workshop a few summers ago. So I've been trying to take care of these guys, and I've also been trying out different mediums in order for them to grow, and I seem to be getting the best results with rainwater and mixed in with some dry grass cuttings, because that does provide raw material. And then, of course, the duckweed do their photosynthesis thing and off they go. But the question comes down to is, how do we know if it's successful? How do they reproduce? We look at the number of leaf blades that they have, and we can count. And that's what our data is going to be. We're going to look at how well do these guys here in different concentrations of Coca-Cola and water. You'd say, well, wait, Coca-Cola, the soft drink? Right. Well, what's up with that? Let me adjust the camera here, because now you guys got to see the duckweed. And now I want to show you guys what I'm talking about. If you look here under the ingredients, you would find... let's see, carbonated water, high fructose corn syrup, caramel color. That's what we want, phosphoric acid. Okay, so we're going to measure the concentrations of phosphoric acid in these cups here, and we're going to grow our populations. Okay, we're going to run the experiment, I guess, for maybe about a week or so, and we'll check out the results and graph the results. So this is going to be a unified project for AP Environmental Science and my regular chemistry classes. Simply because we're talking about concentration, and what a great opportunity for an AP class and a regular science class to work together on this. Okay, so what's going to be involved? Well, we got the duckweed, we got our phosphoric acid. Now the Coke has long since been expired. It's like October 07. Okay. We don't care about the expiration date. What we want, it's the phosphoric acid. Now some people might say, but you know what, you have all those other ingredients in there. Won't that affect the outcome of the experiment? Well, there are limitations. I don't have phosphoric acid available except in this, but I want to try it. So there are limitations to the experiment. Okay, so this is a way of looking at a toxicology study for AP Environmental Science and looking at the effects of concentration on different things. So we're going to measure the number of leaves on leaf blades on our duckweed, and thus we can get an idea of the population count. Okay, a lot of info for the first few minutes. All right, so what do? Well, I measured out cups here, okay, and I measured 40 milliliters of liquid that can fit into these cups. All right, so I got 40 milliliters of Coke, 0 of water. Okay, now I have the other end where I have 0 milliliters of Coke and 40 milliliters of water. Now the water is going to be regular tap water. I'm going to keep it simple for that. Well, actually it should be purified tap water. Okay, now I have 30 milliliters of Coke, 10 milliliters of water. I have even concentration of Coke and water, even volumes. Okay, then I have 10 mils of Coke, 30 mils of water. So that's the idea, and then of course 5 milliliters of Coke and 35 milliliters of water. So that's the idea about the experiment. We're going to fill those up with different concentrations of Coke and water. Then we're going to take these duckweed here, these aforementioned guys, and we're going to put them in there, and then we're going to have an initial count, and then we're going to set them off, okay, to a place and leave them alone. Now I'm going to have to make sure the wind doesn't knock them over or whatever, but that is the general idea of the experiment. We're going to see over a period of days, every day, I'm going to take some pictures and post up the data. All right, let's see what happens. Okay, so I went ahead and I mixed different amounts of Coca-Cola and purified tap water, at least using the Brita water pitcher with filter. Okay. So this one here is all Coke, no water. And this next one is 30 milliliters of Coke, 10 milliliters of water. You already can see a little bit of a difference in the coloration. Here it's 20 and 20. Again, you can look at that. Then you have 10 milliliters of Coke, 30 milliliters of water. And again, notice the caramel color is now becoming less and less. Then there's 5 mils of Coke, 35 milliliters of water. And we've got absolutely no Coke, all water. So you guys can now see the difference in the concentration of the Coca-Cola with the water. All right, now the next thing is getting in those little duckweed. All right, we measured out the amounts of water and Coca-Cola. I went ahead and I selected the greenest duckweed that I could find, and I put a number of them into each of these things here. Now, they're out in the sun at the moment, and the next question comes down to is, well, how am I going to take the data? What is the data? Why are we doing this? Okay, we want to see the effects of different amounts of phosphoric acid on the growth of these little green guys called duckweed. You don't find them in California, you find plenty of them in Texas, and they're very easy to grow, and we can test out different things. This is one way that we can do, we can study toxins, or poisons if you will. So, on this group here, you can see that we did it a range of 40 and 0, that's Coke to water, 30 and 10, 20 and 20, and then when we get down to here, okay, it's 10 to 30, 5 to 35, and then 0 to 40. So we run the scale on that. So what is our data going to look like? That's the key. We're going to be looking at the number of blades that they count. So let's take an example. I'm going to go up to here, and I'm going to go to this one here. This is the 3010. All right. And I'm going to count, and you notice that up here I've got 2 blades. So that's 2 so far. I've got here 3. I have another one here off to the side, that's 3, and then 2, and then here I got 1, 2, 3, 4, 5. The non-green one we don't count, so there's 5 and 3, 8, 8 and 2, 10, 10 and 3, 13, 13 and 2 is 15. So our starting data for this cup is 15. So that's how you guys are going to count, okay? And each day I am going to expose them to the sun for a little bit so they can do the photosynthesis thing. Okay, but I'm also going to cover them up in this box, okay, to protect them from the wind. So I'm going to go ahead and I'm going to start with this one here. So I'm going to have you guys go ahead and count how many blades. Pause the video and go ahead and count. Okay, then I'm going to go back to here. So here's 30 and 10. Pause and count. Here it is 20 and 20. get a little closer here. Again, pause and count. Alright, as we move a little more out here in the sun. So this is 10 and 30, 10 Coke, 30 water. So again, pause and count. Even if it's a little tiny green thing, count that. Okay. And our next one here is 5 mils of Coke, 35 of water. Pause and count. Okay. And then this last one, which is our control. We've added no Coca-Cola to this whatsoever. Now remember, there are other ingredients in the Coca-Cola, that is true. In this case here, ladies and gentlemen, we are interested in seeing what the effects of phosphoric acid is in there. That's what I'm really looking at. The sugar is long since broken down. This Coca-Cola is spoiled. But it still smells like Coca-Cola. It still has the coloring of Coca-Cola, sort of. Okay, but we want to find out. So this is day 1. Okay, and I'm going to take photos and I will put those up on Sarge. Alright guys, I will catch you later. Let's see what happens with this experiment.